Hey everyone, my name is Dr. Raghav Sehgal, and I am a research faculty at Yale University and the lead author for the publication Epigenetic Aging Biomarkers Are Responsive. And as you might know, in the study, we have looked at close to 51 different putative longevity interventions with pre and post DNA methylation to look at responsiveness of 110 different DNA methylation aging biomarkers. And to do this, we, of course, curated and harmonized a lot of these data sets from publicly available and some private sources. What I want to do today is show you how you can access all of this data that we have curated and harmonized over the last 2 years and more, actually, and access it through our platform called TranslAGE. So to give you a quick brief about TranslAGE itself, TranslAGE is a platform that has been used to evaluate the prognostic responsiveness and stability or reliability of aging biomarkers in general. And this is sort of the front page of this website, of this dashboard, which is called translage.io. And you can access this website from anywhere. Just sort of search translage.io in your browser and you will land on this page. And we have not just the data sets that we have in this Nature Medicine study that is coming out, which are the 51 interventional data sets, but also a lot of additional data sets on this platform, and keep on expanding them. And I'll talk about that very briefly as well. Though we have a separate publication talking about TranslAGE as well, and I will direct you to that for further reading. As I mentioned, the TranslAGE website, or the framework in general, is used to evaluate the prognostication, the responsiveness, and reliability of aging biomarkers. And you can do this via the STAR framework: association, risk, treatment response, and stable. Again, go ahead and explore this, and as well as read about it in our TranslAGE publication, which is currently a preprint on bioRxiv, and I can put the link of that in our YouTube video. But more importantly, I want to give you tell you how you can access the data sets for this Nature Medicine publication, where we have put in all of these 51 interventions. So to do this, what you have to go and look at is this Explore Our Resource page, which will show you all the data sets that are currently harmonized and curated and available for you to use on TranslAGE. These are about 179 data sets from publicly available and some private resources, mapping to about 42,000 individuals, human samples. Let me reiterate that. These are primarily blood-based samples at this point, but we are expanding into other domains as well. And again, this is all DNA methylation data. And you can subset this data in different types. You can see, of course, all of this data has been listed here at the bottom, but you can subset this data in different types: events or conditions, so on and so forth, based on what you're interested in: longitudinal, cross-sectional, different analysis pipelines that we have, for example, treatment response, associations, risks, stability, which is the reliability. And that's another publication that we have as a preprint, which you should definitely check out. And you can look at all of these in our research collaboration, research sort of section here. But today I want to specifically talk about sort of the Nature Medicine publication and the data sets available in it. So when you click on Sehgal et al. Nature Medicine, which is currently in review and hopefully going to be published very soon, when you're watching this video, this publication should already be out there. You can see this is subsetted to data sets that are that are currently available and in this particular study, or used in this particular study. Each intervention that was used, for example, the Green Mediterranean diet, it has a public ID, which is, you can literally sort of pick this public ID, and this matches to sort of an EMBL ID, which is one of the databases that we use to look at these. And just Google it and you'll find the particular data set that we used in this case, and you can look at it, the raw data and all its information. But we have also provided sort of a short description, which you can use it to understand this particular data set or intervention better. And so there are some EMBLIDs as well as some geo data sets. So if different sort of types of data sets have different IDs. But most importantly, what we have done, we have also sort of given some information about the number of samples and arrays, but most importantly, which I wanted to talk to you about, is that we have made all of the data that we use to generate and sort of all the harmonized data sort of available to you with very ease, with a lot of ease. So this download data button will essentially sort of lead you to a linked page where you can download all of this data. Note that this has two files in each of these folders. This is called the resid file, which has the actual sort of the calculated DNA methylation values for all the clocks that we used in our analysis. And these might have more than some of the clocks that were used in the original Nature Medicine paper because we're continuously updating a lot of the clocks. So this would be a lot more potentially clocks and epigenetic biomarkers. So more data for you. Be excited about that. And then, of course, we have the harmonized data, which is the raw DNA methylation values mapped to the phenotypic data. And what we have really done is sort of map the same phenotypic columns across these different data sets, such as sample ID, age, sex, response time, so on and so forth, so that people can really run scripts across the board, these different data sets, and have like harmonized scripts. And so I'm going to sort of, I've already downloaded these. As you can see, these are our data files. So they would be super easy to open in something like RStudio. And I'll just show you what these files actually contain. So as I mentioned, we have the original resid files, as well as the the cleaned raw data file. So let me just quickly open the resid file, as well as, while that is opening, also open the clean file, which takes a little longer to load. So the resid file, as I mentioned, is has these standardized columns you would see: sample ID, subject ID. So sample ID is typically the sample ID that is there given from EMTab, sort of EMBL, or from GEO, which we use for an independent identifier. Subject ID is because there are multiple subjects in this study, right? Which might have which might be sort of tracked longitudinally. So you want to track them with the same subject ID. So for example, this particular subject 4 has 2 samples taken longitudinally, pre and post the intervention. And then each of the ages, pre and post the intervention, what they're sort of, whether they are female or male, the group that they lie on, sort of, and the C time. Now, this is really important in case of the intervention. This is the period of the time the intervention ran. So, and this is measured in days. So it's 0 to 540 days. So this sample was taken at 0th day. This sample was taken at the 540th day. So as you can see, it's an 18-month long intervention. So fairly long intervention. We also keep the original sort of columns that were there in the EMBL file in case the user wants to know what other information is there. You can see all of this data here as well. But more importantly, what's really exciting is you also have, we have curated all of the data and clock values that we have. So for example, we have Hannum, Horvath, Thorvat2, PhenoAge, DNA MTL, cause age. All of these clock values sort of calculated and kept here in a single file, which would be pretty easy for you to access. So that is one thing that I wanted to share with you. And this is one of the basic files. And you can see we have a lot more epigenetic biomarkers here because we have really expanded it into a lot of epigenetic proxies and stuff, which you can now access really quickly. And as I mentioned, we have calculated a lot of sort of this data already for you, and you can really access it very quickly. Now, the second file, of course, is which I'd uploaded is the large DNA methylation file. I'm not going to open this file because it's pretty huge, but this contains all the samples in rows, and in columns, it has sort of, since this is an 850 EPIC V1 array, about 8,000 or something, 800,000 something CpG DNA methylation values per sample. So it's a fairly large data set. I would say if you look at it in size, I would say this is, if I change it to the grid format, you'll see this, it's 1.1 GB in size. So it's a fairly large data set. And then, of course, we have the phenotypic file, which has the same sort of C sample ID, C subject ID, C female, and C group, and C time, but also has all the other phenotypes that I had initially mentioned, which were used in the resid column. So if you really want to sort of replicate our analysis in any way, you can go through the DNA methylation data, use the metal cipher package to calculate all the clocks that we have calculated, and then get to sort of this DNA MH file very, very quickly, which is in all honesty pretty amazing. And we have some sample scripts for the DNA methylation for metal cipher already available on the metal cipher file package. And so yeah, this is how you can use this data with a lot of ease. I also wanted to quickly show you how you can actually generate some of these plots that we had in our publication in the treatment response dashboard. So of course, we have the association and the risk dashboard and the stability dashboards and everything. But I did want to talk about that which you can explore if you'd like, which have association analysis with future prognostic outcomes, and stability, which is around technical and biological stability. But I did want to talk about sort of these biomarkers that we have here. So as you can see that you can subset these data sets, we have a lot more interventions now because this is an ever evolving database. We have actually gone from 51 longitudinal longevity, putative longevity interventions to close to 103 now. So we're really sort of expanding this database. So if you want to just sort of subset to the original that we used in our study, you can just sort of use this button and use the Segal et al. 2025. And then you look at any of the biomarkers that you're interested in. So for example, all the mortality clocks, the famous ones, such as GrimAge V1, PZ GrimAge, systems age, all of its components, so on and so forth. You can all sort of look at them. How are those clocks sort of specifically fluctuating, in the sense that are they responsive to interventions or not across the board? You can give that answer very, very quickly, which what we had done in our paper. You can get this in a second here. You also have sort of the individual data sets and if they're responsive across the dashboard for this. And what's most important is you can download all of these as well, if you like, in a very quick and easy format. And so again, I just wanted to put this out there for you to leverage it and use it in your analysis. You can download the CSVs, PDF. All of this data is accessible, usable for you to analyze, leverage. And I'm not going into a lot of detail today about the other parts of this dashboard. We have a couple of other publications, as I mentioned, coming out. So we have the TranslAGE publication, where I'm one of the co-first authors, which you can read on bioRxiv. We also have sort of the biological versus technical reliability publication, which talks about the stability of aging biomarkers. And so I would definitely recommend that that you check out these papers and gain more insights about what we're doing with the TranslAGE dashboard. But most interestingly, you can leverage all the data that we have here in an easy, quick format. And just to remind you, all of this is sort of simply accessible through here. I will also point out one last thing, that some of these data sets are sort of protected data sets. So for example, if these are data sets that were accessed by sort of built by TrueD or were generated by TrueD, this download data form will essentially direct you, this download data button will direct you to a form which you can fill out to access data via TrueD. And we will be sharing TrueDiagnostic, sorry, I meant TrueDiagnostic. And you can access this data from TrueDiagnostic by requesting it along with the original authors of this data. As I've said, we've really made this data set really sort of all the data that we have used really accessible, and hopefully it can be a powerful dashboard and tool for everyone to use in their future analysis. Well, thank you so much, and I really appreciate you listening me out, and I hope to talk to you in the future about this really amazing platform that we have built and this very interesting study we have done. Thank you.